All right, welcome everyone. My name is Megan McGrath, and I am the Education Programs Manager here at the Duke Leamer Center in Durham, North Carolina in the United States. But we are hosting a series of Malagasy scientists, and we're very excited to learn today. So I will go ahead and pass it over to our partner in Madagascar at CURSA, Marie Roland.
Thank you, Megan, for this presentation. So, hello, everyone. My name is Marie Roland Swazafi. I'm a teacher at CURSA. And today we are happy to welcome our speaker, Miali Razanajatovo. She's a Malagasy ecologist. And we are very proud of you, Miali. And she's a mother of one child. And she published 15 papers in international journals. And she received different awards and grants, including a grant from a German Research Foundation. And she studied in the University of Antananarivo in 2009, and she got her master's degree in the botany department at this university. And in 2011, she did her internship at the Institute of Plants and Science of Plant Sciences in the University of Bern in Switzerland. And then in 2016, she got her PhD in ecology from the University of Constance in Germany. Then from 2016 to 2021, she did her postdoc in the ecology group at the University of Constance in Germany, always. And since 2021, she becomes a senior researcher associate at the Institute of Landscape and Plant Ecology of the University of Hohenheim in Germany. Wow. So thank you for joining us today. And also great thanks to the DLC for hosting this virtual talk. And a great thanks also to the Duke Africa Initiative for supporting this. Thank you, Miali. So welcome. We are listening to you.
Thank you very much, Marie Roland, for this very kind introduction. I very much appreciate the opportunity to talk about my work with you today. So let me share my screen. Now I hope everything's working. So today I'm going to talk about indirect interactions between native and non-native plants via pollinators. But to start with, I would like to tell you how I got interested in invasive plants. So this is when my interest in biological invasions began. Some years ago, I worked as a field assistant for a team that studies seed dispersal by lemurs in Manum Reserve in Farfangan. And we observed our lemurs a number of times sitting and feeding on the fruits of this introduced tree species. So this is native to the Americas, and it's considered as one of the worst invasive species in the world by specialists. So I became interested in why some species like Cecropia become invasive and how do they form non-historical interactions with new species in the invaded range, like with the lemurs, with which they did not share any evolutionary history with. So these pictures show shipping lines and airline routes that connect regions that are isolated naturally due to biogeographic barriers such as oceans and mountain ranges, but now they are very much interconnected via shipping routes and airline routes. And so by moving around the globe, humans have contributed to the introduction of non-native species in most parts of the globe. In parallel with the development of shipping lines and airline routes, the introduction rates of species have tremendously increased across time. And so this is, for instance, the first record rate of vascular plants from the year 1900 to 2000. And we can see that the number of introduced species has accumulated across time. But it will continue to increase according to predictions. So this is also for vascular plants and projections in different biogeographical regions. So it is, to me, it is important to understand what determines invasiveness for at least two purposes. So first, for management and conservation. And second, we can use biological invasions as natural experiments to understand ecological processes. My approach is twofold. I look at the characteristics of the alien species and also look at the characteristics of the invaded native communities. And I focus on reproductive characteristics. I will elaborate more on this in the next slides. So I look at plant pollinator interactions, floral traits, and the selfing ability, for instance. So today's talk will be organized in three main parts. So in the first part, it will be an introduction to the importance of reproductive characteristics in plant invasions, and they're highlighting some of my previous work. And in the second part, I will report on a recent experiment on facilitation and competition in pollinator-mediated interactions. And in a third part, I will report an experiment on pollen interference among alien and native species. The invasion process is a series of barriers and stages from the introduction of a species to a non-native region to becoming invasive in this region. So once a species manages to overcome the biogeographic barrier, so as I mentioned, oceans or mountain ranges, we call them alien. And once an alien species manages to found and maintain reproducing populations in the wild without further human intervention, we call it naturalized. And once naturalized species manage to disperse and spread in the new regions, we call it invasive. So now, I would like to stress the importance of the reproductive barriers to becoming naturalized. So reproductive barriers are important. They play a very important role in naturalization and invasion, because naturalization is a key stage, as all naturalized species are potential invaders. But in general, to what extent do pollinators contribute to seed production in plants? So in this global synthesis that we recently published, we compiled databases on pollination experiments and quantified pollinator contribution to seed production. So to calculate the degree of pollinator contribution, so PC here, we compared reproduction under natural conditions and reproduction when pollinators are excluded. So in this frequency diagram, you can see, you have PC value of 0, which means that the plants are, so pollinators do not contribute to their pollination. Pollinators do not contribute to their seed production, and this includes the wind-pollinated plants, whereas pollinator contribution of 1 means that pollinators contributes to 100% of seed production. The phylogeny here shows the distribution of our data across the angiosperms, and the map here shows the distribution of our data across the globe. So what we learned from this is that about half of flowering plants relies on pollinators for at least 80% of their seed production. So how do alien plants reproduce when they are introduced into a new region? Most plants are hermaphrodite, so they are technically able to fertilize themselves. So we call this selfing. But due to different disadvantages and advantages of selfing, the degree of selfing ability differs among plant species. So we calculated a selfing ability index by comparing reproduction after hand pollination using self-pollen and hand pollination using cross-pollen. And we used a path analysis to disentangle direct and indirect effect of selfing ability and naturalization. So selfing species with high selfing ability are more likely to have large native range size, and in turn, species with large native ranges are more likely to become naturalized somewhere. And then when we look at the species that are already naturalized somewhere, we found that the degree of a naturalization extent, so the number of regions where a species is naturalized, was directly related to selfing ability of species. So we concluded that a lack of mates and pollinators in the new region may be a barrier to naturalization of species lacking selfing ability. So alien plants are often decoupled from their historical pollinators, and the ones that can attract resident pollinators should have an advantage. To compare flower visitation among native, naturalized and non-naturalized species, we used the Botanical Garden of Bern in Switzerland. So the botanical gardens represent great opportunities to do multi-species comparative studies, because species from different geographic origins are grown under similar conditions. So we found that the species that are grown in the gardens but did not manage to become naturalized in the wild received fewer flower visitors than naturalized species and native species. So we concluded that non-naturalized plants may be pollinator limited. And in a common garden experiment, we compared pollen limitation of invasive and non-invasive naturalized alien species to native species. And to calculate the degree of pollen limitation, we compared reproduction after pollen supplementation and reproduction after natural pollination. So a value of 0 for the pollen limitation index represent low degrees of pollen limitation. So we found overall a low degree of pollen limitation, and there was no difference in the degrees of pollen limitation among native and naturalized plants. So to sum up this introductory part, we learned that alien plants can form novel interactions with resident pollinators. And in the rest of the talk, we will talk about how alien plants receive pollination services in the invaded range, and whether alien plants suffer from native heterospecific pollen. And finally, whether phylogenetic relatedness and trait similarity between alien and native plants play a role in the outcome of interactions. The first study that I'm going to talk about is on the pollinator facilitation and competition for pollinators in alien and native plants. I did this study together with Phelan and Prensi from Madagascar, who came to work with me in Germany with the support of the DAAD scholarships. There are two hypotheses with opposing predictions on how relatedness between alien and native species should affect naturalization success. So according to the pre-adaptation hypothesis, the alien species that are closely related to the native species should be more likely to naturalize because they are pre-adapted to the abiotic environment. So on the other hand, according to the Darwin's naturalization hypothesis, because related species are likely to be more similar, they should show strong niche overlap and compete for shared resources. So alien species that are related to the native species should be less likely to become naturalized. So these seemingly contradictory hypotheses are coined the Darwin's naturalization conundrum. And so far, very few studies tested these hypotheses using indirect interactions, such as those mediated by pollinators. So flower visitation to alien species may be facilitated by co-flowering natives if they share similar floral traits. So this show hypothetical plant communities invaded by an alien plant. So if the alien plant is closely related to the dominant native species, then we would expect that they can use the same pollinator communities as the native species. But if they are distantly related to the native species, because of different floral traits, they may only use a small subset of these communities. So in the case of pollinator facilitation, we would expect a negative relationship between phylogenetic distance between alien and native plants and flower visitation to the alien plants. On the other hand, if the alien plant that is closely related to the native species strongly compete with the native species, it may only receive few pollinators, whereas if it has other traits that make them attractive to the other pollinators in the community, it may receive more pollinators. So in the case of competition for pollinators, we would expect a positive relationship between phylogenetic distance between alien and native plants and the flower visitation to the alien plants, excuse me. So to test this hypothesis, we asked the question whether flower visitation to alien plants depend on phylogenetic relatedness and floral similarity to native plants. So in a field experiment, we simulated invaded communities by adding potted alien plants into co-flowering native communities. We used time-lapse video recording to observe flower visitation to pairs of host native plant and the added alien plant. And our field sites were meadows near Constance in Germany. So we selected alien species that covered a broad variation in floral traits, such as size, symmetry, and colors. And they should occur in the grasslands near Constance and usually flowering between April and September. So we pre-cultivated the plants in the botanical garden of the University of Constance. So we had 34 herbaceous species that were insect pollinated. And before adding the alien plants to flowering resident native communities, we prospected grasslands around the city of Constance and identified sites that were visibly dominated by one flowering native species. So for instance, we have this site that was dominated by those purple flowering Lytrum salicaria, and this one site that was dominated by the yellow flowering Lotus corniculatus. So in total, we had 20 host native plant species. So the dominant flowering plants at the site. So each of those species was the dominant native at one or more sites. And we also covered the broad variation of floral traits there regarding size, symmetry, color, and nectar production. So this is to show our study site. So we have a map of Germany here. Constance is at the bottom south part, so just at the border with Switzerland. And our grasslands were mainly around the University of Constance, and the large part of the city of Constance is down here south. So to simulate invaded communities, we placed for up to 10 alien species, up to 5 plants per species. So we left the plants for 3 days at each site before video recording. And in total, we had 151 combinations of alien and native species, which covered phylogenetic distances between 10 and 300 million years. And we did floral measurements on, so we recorded flower size, symmetry, and floral reflectance. And we collected data on nectar production using published databases. To record flower visitation, we placed a time-lapse camera above the flowers of the paired native and alien plants. And we set the time-lapse interval at 2 seconds. We recorded from 10:30 to 16:30 on a sunny day. And by using 40 cameras, we could observe many pairs of plant species simultaneously. So in total, we gathered more than 3,000 hours of observations, which then we processed manually. To process the video, we used the Blender software. So we counted flower visits to the alien and to the native plants in each pair. So in this example, we have the native Asteraceae, Synesio aquaticus, and the alien Papaveraceae, Escholzia californica. So we counted flower visits and identified flower visitors for each 1 hour interval within the whole recording time. And we also counted the number of observed alien and native flowers units within each frame. So we consider the flower visit when the flower visitors made contact with the reproductive organs. And this is an example of a visit on a native flower. This is an example of a visit on an alien flower. Here are some details about data analysis. So we used a phylogenetic tree of an existing phylogenetic tree of the European flora, which we pruned to match our alien and native species. And then we calculated a phylogenetic distance between each pair of alien and native species using the ape package in R. And so the floral traits that we analyzed were flower size, symmetry, reflectance in the different visible wavelength, and nectar production. So we calculated an overall floral trait distance using the Gower dissimilarity in the FT package in R. And we also calculated single floral trait distances, so both absolute distances and hierarchical distances, which took into account the direction of the differences or distances. In the statistical analysis, so we wanted to answer the question whether flower visitation to alien plant species depended on the phylogenetic and floral trait distances between the alien and the native species. So we used 3 response variables: the number of visits to the alien plants, the proportion of visits to the alien plants relative to the total of visits that alien and native received, and the similarity in composition of flower visitors to alien and native plants. The explanatory variables were phylogenetic distance between the alien and the native species, and we included a quadratic term to test for U-shaped or hump-shaped relationship. We also included as explanatory variables the floral trait distance between the alien and the native species. They're also including a quadratic term. And finally, single floral trait distances between the alien and the native species. To account for potential variation in flower visitation due to floral characteristics of observed plants, potential variation of flower visitor activity, identities of alien and native species, we included further explanatory variables and random variables. So, just to remind you about our predictions, negative relationship between the phylogenetic or floral trait distances and flower visitation are expected for facilitation, and a positive relationship is expected for competition. But what we found were, so now on the first graph, you have on the x-axis the standardized phylogenetic distance between alien and native plants, and on the y-axis, the number of visits to the alien. So on the second graph, same, but we have with floral trait distances. So for the phylogenetic distance, we found a U-shaped relationship. And interestingly, for the floral trait distance, we found an hump-shaped relationship. So the number of visits to the alien plants were lowest at intermediate phylogenetic distances and highest at intermediate trait distances. So when we look at the models with the proportion of visits, we found similar patterns with U-shaped relationship with phylogenetic distance and hump-shaped relationship with floral trait distance. When we look at the similarity between alien and native plants, we found a negative relationship between phylogenetic distance and this similarity, but U-shaped relationship for floral trait distance and similarity. So the least similar flower visitor composition were at large phylogenetic distances and at intermediate floral trait distances. So what does all of this mean? We interpreted this, so the nonlinear patterns that we found between flower visitation and similarity as an interplay of facilitation and competition, so acting simultaneously. We found a discrepancy between the effect of phylogenetic and floral trait distances, and this is probably due to different strength of phylogenetic signals among the traits that we used. And so the effect of single trait distances were also different. So for flower visit, for instance, for the absolute distances, flower visitation decreased with flower size distance. in dissimilarity in reflectance in the blue wavelength, which is in line with facilitation. But flower visitation increased with dissimilarity in flower symmetry and with the reflectance in the green wavelength and nectar production, which is in line with competition. And when we looked at the hierarchical distances, we, for instance, found that flower visitation to alien plants were higher when native plants had reflectance pattern the blue wavelength, but the alien plants did not. And flower visitation to the alien plants were also lower when the native plants produced nectar, but the alien plants did not. So to sum up, this study showed that novel plant-pollinator interactions could be driven by an interplay of facilitation and competition, and floral similarity or dissimilarity plays critical role in pollinator-mediated interactions between alien and native plants. And finally, patterns for phylogenetic and floral trait distances do not need to be consistent, and they can reflect different mechanisms. And now, what happens post-pollination? So the second study that I would like to share today was led by Eva, who was then a PhD student co-supervised by myself. And I would like to highlight that Eva went to Madagascar in 2018 to teach R and stats within an event organized by the association ecology at the University of Antananarivo. So I'm going to talk about heterospecific pollen interference. So what is this? So when plants are co-flowering in communities and sharing pollinators, they are exposed to heterospecific pollen. That means pollen from species A can be deposited on the stigma of species B via a shared pollinators between those two species. And the interference can occur at different levels on the pistil of the heterospecific pollen recipient. So at the stigma level, heterospecific pollen, so the foreign pollen, can inhibit pollen at the adhesion or hydration or germination of the right pollen, so the conspecific pollen. At the level of the style, there can be a mechanical cloaking of the conspecific pollen, or even an allelopathic inhibition of the pollen tube growth. And at the level of the ovary, heterospecific pollen can try to fertilize the conspecific ovules, resulting in ovule abortion or non-viable hybrids. So in most cases, heterospecific pollen interference is leading to reduction in fruit and seed production. So there's been a meta-analysis that looked at the effect of heterospecific pollen deposition on native pollen recipients by native and alien pollen heterospecific pollen donors. So what this meta-analysis found was that overall, heterospecific pollen had negative effect on the reproduction of natives. And when the heterospecific pollen donor was alien, the effect was more negative than when the heterospecific pollen donor was native. And also, when the heterospecific pollen donor was related to the native pollen recipient, the effect was more negative than when the pollen donor was not related. So we were wondering what happens to the aliens. So is there anything, there was nothing known about the the effect of native heterospecific pollen on alien plants and the effect of heterospecific pollen from other alien plants on the alien plants. So in a greenhouse experiment, we asked if alien species also suffer from pollen interference from the natives and from the other aliens. So we applied pollen mixture on the stigma of recipient plants. We had 2 pollination treatments to simulate heterospecific pollen deposition. So a control treatment that consisted of 100% conspecific pollen, and a heterospecific pollen treatment that consisted of 50% heterospecific pollen and 50% conspecific pollen. We had a total of 20 species, so 11 species native to Germany and 9 species aliens in Germany. And we tried to have all possible combinations of alien-alien, native-native, alien-native, and native-alien heterospecific pollen donor and recipient species. So we achieved 155 species combinations with 1,700 flowers treated. A major part of this experiment was done by Sylvie Berthelot, who was then a master's student supervised by myself. So here are some details about data analysis. We did the same thing to calculate phylogenetic distance between excuse the typo here. What matters is the phylogenetic distance between pollen donor and pollen recipient species. And now we analyzed pollen size and style length as floral traits because these matter most in pollen and pistil interactions. And we calculated single floral trait distance, which was a signed distance that also accounted for the direction of the distance. So we asked whether heterospecific pollen affected plant reproduction and whether this depended on the alien and native status of the pollen recipients and the pollen donor species. So our response variables were fruit set and seed set. And we used linear hypothesis testing and multiple comparisons with a custom set contrast matrix. So we found that alien plants suffered from pollen interference with the natives, but not with the other aliens. So let me guide you through the graph. So on the first graph with a fruit set, so you have on the x-axis the pollen recipients. So blue is for native and red for alien. And then the different treatments, whether it was a conspecific pollen treatment or heterospecific pollen treatment. So now for the heterospecific pollen treatment, when the donor was native, so the blue dot, then heterospecific pollen significantly reduced fruit set. Same for when the pollen donor was alien. So when pollen donor was alien and pollen recipient was native, there was a significant reduction in fruit set compared to conspecific pollen. So here, regardless of whether pollen recipient was alien or native, heterospecific pollen generally reduced conspecific pollen, generally reduced fruit set compared to conspecific pollen. And now, when the heterospecific pollen recipient was native and the heterospecific when the heterospecific pollen recipient was alien and the donor was also alien, then there was no reduction in fruit set. So this seems like there's a facilitation of the other alien species. And we term that invasional meltdown. And last, when heterospecific pollen recipient was alien and the heterospecific pollen donor was native, then there was a significant reduction in the fruit set. So it could be that there is a biotic resistance mechanism of the native communities. When we look at seed set, the only significant relationship that we found was that generally heterospecific pollen decreased seed set compared to conspecific pollen. We did not find any effect of phylogenetic distance on pollen interference, but we found some effect of floral trait similarity. So first, a similar style length increased heterospecific pollen interference. So in this graph, on the x-axis, you have the style length distance. So negative distances means that the style length of the recipient species is longer than the style length of the donor. And the value of 0 means that style length of both donor and recipients are similar, and positive values means that the style length of the donors is shorter than the style length of the recipient. So what this graph, both graphs show, is that fruit and seed set are strongly reduced at intermediate when style lengths between donor and recipient species are similar. And we found the opposite pattern with pollen size. So a dissimilar pollen size now increased heterospecific pollen interference. The same, you can read the x-axis in the same way. So these are negative values are for pollens. Pollen of donor is larger than pollen of recipients, and positive values, pollen of donors are smaller than pollen of recipients. But overall, fruit set and seed set are strongly reduced when pollen size between donor and recipients are dissimilar. So what did we learn from this study? Both alien and native recipients suffered from heterospecific pollen from donors of the opposite stages. And phylogenetic distance did not affect fruit and seed set. The style length distance decreased heterospecific pollen interference, and pollen size distance increased heterospecific pollen interference. So these are a lot of information. But from this presentation, I hope that you learned that floral similarity can predict the outcome of indirect interactions between alien and native plants. And with this, I am at the end of my presentation, and I would like to thank my collaborators, my assistants, and the funding agencies, and of course, all of you for your attention.
Wonderful. Thank you so much, Miali. Do you want to go ahead and stop sharing so we can see you as we ask questions?
Yes.
Okay, great. And we have a few questions for you. I have a few questions for you, learning. And I'm sure we'll have more. And one of our first ones, actually, I know you were just speaking about all of your research in Europe, but we did have someone who was wondering about Cecropia in Madagascar, because that was new information to them. And just, do you know any more about its distribution and impact from your time studying the ruffed lemurs?
Unfortunately not. For the outcome of that study I was involved in, we did not find, I think, if I remember correctly, we did not find evidence that the lemurs, which were Variesia variegata, did not, so we didn't have evidence that they dispersed the seeds because we didn't find like mature seeds in the feces of the lemurs, probably because they were feeding on unripe fruits. I'm not sure, but unfortunately I haven't so far seen any other studies or interest in distribution and dispersal of Cecropia.
Okay, no problem.
I'm wondering if this is also spreading in the northeast now. I don't know if Marie Roland or the team in Madagascar are aware of that. So that was 13 years back that I was in Farafangan.
Right. Okay, that's no problem. We have plenty of questions about your research too. I will check in. Marie Roland, do you have anything to add or any questions from your group?
Yeah, I just discussed with Christophe here, our director, and he said that lemurs can be both pollinator and also predator. So yeah, so it's not yet sure, just like what Miali said. Yeah.
And it sounds like this is something maybe some of the students watching could study and we could find out more.
Yeah, okay, thank you. Yeah, and also we have a question because we have our vanilla, which is still not yet naturalized until now, since there is no natural pollinator until now. And then it's a hard work for our farmers, but vanilla production is the main crop of Madagascar. So we need your recommendation as an expert. Since we have native vanilla madagascariensis, and many more native species, what should we do to conduct the same experience, or what is your recommendation?
Yeah, I can imagine that it is a lot of work doing the hand pollination by hand. On the other hand, I would be very cautious if in considering introduction of pollinators. I wonder to what degree there is a similarity between the floral traits of the vanilla crop and the wild species that we have in Madagascar. I am not sure of the evolutionary history. So are they rather closely related or distantly related? And whether we should, I don't know, did you suggest whether we should do similar manipulative experiments to find out? I think it's a good idea. I wonder to what extent you can. How long does it take for the vanilla crop to flower? You know, because like we had those the manipulations were that we brought like plants in pots toward the sides, but maybe we can think about a different design and look at pollination.
Okay, thank you. From the observation, they have like similar flowers, very similar as a flower trait. So this is already very similar. But vanilla takes 1 year for flowering. So it gives flower every year. So maybe the experience will last longer than expected, and yeah, I understand. It could be more difficult than with herbaceous plants.
Yes, but there should be a way to design the experiment so that we still have like two plants flowering that we could expose to pollinators and see the outcome.
Wonderful. It sounds like we're already talking about potential future experiments based on what we're learning. And you've talked a little about insects when we talk about vanilla and being cautious about introducing any insects. And you've talked a lot about alien versus native plants. Did you do any study into whether or not the insects visiting were alien or native, or were you mainly focused on the plants?
Yes, I'm mainly focused on the plants, but also in the temperate grasslands that I'm working on, there's not much of a problem of alien insect species. So most of the species, not to say all of them, are natives. Yeah.
Wonderful. And obviously your study focuses on flowers and plants. Did you get to learn more about any really cool insects while you were doing the study, or work with any entomologists while you were collaborating?
Yes, so I discovered, so during all the studies, I discovered that there's a tremendous diversity of insects that I wasn't aware of. Yes, so it is really incredible how many, I'm not sure anymore about the numbers now, but I think for Switzerland alone, it's like 6,000 species of arthropods, and 60% are visiting flowers. Yes, so
yeah,
what I would like to delve into in the near future is nocturnal interaction. So there are also nocturnal visitors like moths that are really cool here in the temperate grasslands, and yeah, I would love to also learn about them.
Yeah, that would be fascinating. And it looks like maybe we have another question from Marie-Roland and Karsa. Oh, you're still muted.
Okay, so we have a question from the student. As an expert as you, Miali, do you recommend plant species introduction to Madagascar, and which one?
Yeah, so there are two things. So there was this study published in 2012 that it was a really nice study. They compiled the non-native flora of Madagascar. So I think we already have enough non-native flora. So there are like 1,200 plant species that are not native to Madagascar, but were recorded in Madagascar. I don't think we need to introduce more non-native species. Maybe we, I would like to know more about the motivation of the student, why they would want to introduce more non-native species.
I think their motivation is just like a curiosity, but yeah.
Yeah, so there are non-native species that can cause really large impacts on native ecosystems. So this has been shown in different parts of the world, and it has been very expensive to manage and deal with those impacts of invasive species. So I don't think that it would be a good idea to introduce more non-native plant species.
So I have a question about, obviously, when we look at plants, we have millions of years of evolutionary history as well. And at some point, many plants were not native to where they're currently living. So the seeds arrived, many arrived on Madagascar, like lemurs did at some point. So where do scientists like you draw the line between something that is now considered native or something that is considered naturalized or alien?
Yeah, it's a very good question. Where do we draw a line? I think it is in 1492 when Columbus discovered the Americas. This is where the introduction of species by humans started to develop. And from then on, more species have been introduced to new regions. So this the pace for which the species introduction really differed from the natural dispersal that occurred earlier in the evolutionary time.
That makes sense. More human involvement in the dispersal at that point.
Exactly.
Yeah. All right. So I guess I will see Marie-Rolande. Do you have any more questions before we wrap up? No, I think you've given us wonderful answers and a lot to think about. Thank you so much for your time, Dr. Miali, and to everyone who's watching. I will give Marie-Rolande a moment to maybe talk a little about our upcoming presentations.
Yeah.
I also thank you very much again for the invitation and the opportunity to talk about this.
Yeah, thanks a lot, Miali, for the nice presentation. It was very inspiring for us here in Madagascar. And also thank you to the DLC for hosting, and thank you for Duke Africa Initiative for supporting this virtual talk. Okay, thank you. Bye-bye.